Okay, thank you for your attention. Thank you for your gracious introduction, Chairman. It's my pleasure to share with you my subject, gene expression-based classification and risk stratification of pediatric ALL. And my presentation will be divided into the following four parts. The introduction. Leukemia is the most common childhood tumor and remains a leading cause of cancer deaths in the young. There are about 15,000 new patients each year in China. Among them, ALL accounts for more than 75%. As well known, we currently adopt MSM classification, and there are some limits of this method. At first, it was only offered in a few major medical centers in developing countries. In addition, nearly 50% lack any previously known cytogenetic or molecular lesions. And the most important is that the same subtype may have different response to the same regimens. Now we have our scientific problem. How to solve its problem? ALL has been recognized as a process involving multiple genes and single transduction pathways, and gene expression profile-based classification can reveal the nature of disease. In 2011, came from 11 laboratories across 3 continents, more than 3,000 patients were classified into 18 distinct classes with high accuracy, including 8 ALL classes, 6 AML classes, CLL, CML, MDS, and non-leukemia healthy controls. This study indicates microarray-based classification is robust, but it's time-consuming and labor-intensive with high reagent cost and low sample throughput. So the researchers further adopt a multiplex PCR gene expression profile analysis system, hereinafter referred to as GXP. They diagnosed the small round blue cell tumors, prostate cancer, colon tumor, and so on with this method. In this research background, our lab conducted a related previous study, developed a microarray-based subtype classifier based on 62 macrogenes, which can predict 7 different ALL subtypes with an accuracy as high as 97%. Then we extend from microarray technology to GXP technology. It was also called Advanced Fragment Analysis System, or AFA for short, with the minimization of macrogenes. The classifier has a prediction accuracy of greater more than 94%. And the related study also attained the Chinese invention patent last year. Okay, let's move to the 4 part, the method. Advanced fragment analysis only need a PCR sum cycle instrument and a GXP machine. And the principle of AFA technology is that chimerical primers were used when RT and PCR primers. Conversion from gene-specific primers to universal primers can fix the efficiency of the reaction. And all the macrogenes were divided into 3 panels, and B2M, PSMC4, and GUSB were selected for reference genes, and at the same time, cancer-resist gene, pcDNA as quality control genes. When we design the primer, the size of gene must be controlled between 150 to 350 BP. As shown in the figure below, every peak location represents gene identity and peak fluorescence intensity represents gene expression level. We enrolled 188 patients from August 2007 to January 2014 and diagnosed as AL based on MSM classification, treated them according to BCH 2003 or CCLG 2008 protocol. January 1st this year as the end of collection of the patient treatment outcomes. Okay, let's move to the result. The risk group of 188 patients according to MSM classification and corresponding EFS rate are shown in this slide. The most of them were classified into the intermediate risk group, and the overall EFS rate exceeded 82%. This figure shows the hierarchical cluster of AFA samples. We can see every subtype clustered together very well. The subtype of all patients according to AFA classification and corresponding risk group are shown here. We just relied on the subtype, also classified them into 3 risk groups. And the EFS of every risk group has a significant disparity. Now let's move to the fourth part, the discussion. AFA technology combines RT-PCR and capillary electrophoresis to deliver a simple and effective multiplex assay. And this technology only needs 1 day and $100. But MSM classification often need 5 to 7 days and $500. The 5 years EFS for every risk group, according to the new risk stratification, shows a significant disparity, and AFA classification has a certain prognostic value and can be further discussed. So we will enlarge the samples to validate its accuracy and follow up all the patients to further observe its prognostic value. At last, acknowledgement. The members of our hospitals design and implement the whole studies. The researchers from Shanghai Institute for Biological Sciences provide bioinformatic analysis for this study. The workers from Health Gene Technology company give support on environmental technology for this study. Okay, that's all. Thank you for your attention and looking forward to your valuable advice.
Thank you. Are there any questions?
The question is they use this in the clinical trials?
Yeah. Do you want to ask a question?
For example, on question, have you used your new risk stratification implement into clinical trials, national-wide clinical trials? Will you use it?
And in our new risk stratification, I only relied on the subtype and BCI.
I mean multicentral.
Yeah, and our hospital, according to MSM classification, we relied on the WBC, white blood cell, and new diagnosed age, MRD, and so on. And in this new risk stratification method, we only relied on the subtype because the new risk stratification method can, such as the other subtype, the other groups can be classified into BCR-ABL or TLML1 according to its actual risk group.
The next.
Hello. So I wanted to ask one thing. This is a known, risk groups of cytogenetics. So basically, you highlighted just a new, you may say, methodology of advanced fragment analysis, which gives you early results. But you haven't highlighted here what's new in this classification. There's no data on the copy number abnormalities or abnormalities in genes like B cell development genes, genes which are involved in cell cycle control and important in B cell regulation. So what is coming up now is an integrated genomics and cytogenetic classification, which is based upon both the results of genome expression profiling and this cytogenetic classification. So what about any results related to copy number abnormalities and any difference in risk stratification?
Sorry, I can't understand.
I think the question is, are you relating your new methodology to a different classification, or are you just describing a new methodology?
Okay. The new risk stratification method is only in the process of research. So it's
Yeah, it's in development. Yeah.
thank you.
I think it's